In this video, we're going to talk about different substances that we need to have in cells and how to get them in and out of cells. And the substances we need can be into two broad categories. Can be put into broad categories: your inorganic and your organic. But organic just means basically made by living things. So organic substances tend to be made by living things, and they tend to all have carbon in their structure. So all organic substances will have carbon in their structure, whereas inorganic substances are not made by living things. And we'll talk about examples of each. So not all of them, but examples of each that we need to have inside cells. The main ones for the organic substances are carbohydrates, lipids, and proteins. Carbohydrates have carbon, hydrogen, and oxygen, so they have carbon in their chain. And they're used for energy mostly, but also they can be found in the form of receptors, or have basically they're attached to proteins to make receptors. We'll talk about that more in a second. Lipids are also organic substances, and they have carbon, hydrogen, and oxygen in their chain, just like carbohydrates, but they just have a different structure. The main lipids you find are fatty acids and triglycerides, and they are used for insulation, so to keep you warm, for protection, because it's basically like a cushion, so you find fat around your organs to keep that from damage. We also have it used as energy, just like carbohydrates. Lipids are the main source of energy in our body. Hormones, so for example, testosterone or estrogen, these are fatty or lipid-based hormones. And then membrane structure. We'll talk about that more in detail, but they're also part of a membrane. Now, examples for lipids would be triglycerides. Right? Examples for carbohydrates would be, for example, glucose is a carbohydrate. Proteins, they have, just like your lipids and your carbohydrates, they have carbon, hydrogen, oxygen, but also nitrogen in their structure. That's the only difference. They basically also have nitrogen in their structure, but they do have carbon like all the other ones. Now, proteins are a major structural component. So what that means is they are found in the structure of most of all cells, and they make up a good chunk of that structure. They also found or are used to make energy, just like with lipids and proteins. Hormones, so we said that lipids have, for example, testosterone, that's a lipid-based hormone. Insulin is a protein-based hormone. So hormones can either be lipid-based or protein-based. And last thing is enzymes. We'll talk about that more in your HSC course, but enzymes basically speed up chemical reactions, and all enzymes are proteins. So those are the three main examples of organic substances we find in living things, and some of their uses. You should know the uses. Now we're talking about a couple examples of inorganic ones. So for example, salts, and the main salts are sodium and chlorine. They're not the only ones, but sodium and chlorine ions are salts. What they help you do is they help you regulate water balance, make sure you've got the right amount of water in your cells and in your body. They also form part of the nerve impulse. So without those salts, we wouldn't be able to actually have our nerves working properly. Calcium ions are found as a major part of the bones and your teeth. So without those, we wouldn't be able to have bone and teeth. But they also have an important role to play in muscle contractions. So muscles only work properly if you have enough calcium, which is usually not a problem, but they need to have that calcium to be able to actually contract. And one more we're talking about is oxygen. So oxygen is needed to make energy. We'll talk about that. It's called cellular respiration. And they are a reactant of that reaction, so they're needed. And those are three examples of inorganic substances that we need to have inside cells, but there are actually quite a few more. So those are just three of quite a few other examples I could have mentioned. So you should know some of those examples and their uses. But what we'll talk about now is how it actually goes in and out of cells. So for example, if this here is your cell, so this whole thing is your cell, here's your cytoplasm, your inside of your cell. Now this here is your outside of your cell. All cells swim in fluid outside that are outside of it. And this is the cell membrane. So the cell membrane would usually be much thinner, but I've just basically drawn it not to scale, but much bigger so I can show you what it's made up of. So this is the cell membrane. It's made up of different parts. So we have your phosphate and your fatty acid tails. So this is your phosphate head on top. So I'll just zoom into one of these here on the bottom. So this one, for example, I'll zoom into this one. It has a phosphate head on top and two fatty acid tails on the bottom. Now the phosphate head is hydrophilic, which means it attracts water. And the fatty acid tail is hydrophobic, which means it repels water, or it doesn't like water. Now that combination is really important because it's going to have a bilayer. It's going to have one on top and one on bottom, and that basically allows it to be able to be selectively permeable. So that means it can choose what it allows to go in and out. What I mean by this is that glucose is actually water soluble. So glucose here, it's a carbohydrate, it's water soluble. And water soluble just means it's attracted to anything that it would be hydrophilic. The glucose is attracted to the phosphate head, because remember that was hydrophilic. But you can get there by being attracted to it, but then it can't get any further, because if it were to try to get all the way through the actual structure, it would bump into the hydrophobic tail on the bottom here, this one, and that would basically not allow it to enter the cell. But we have these protein carriers, which are all around the structure, and these protein carriers can make sure that if we need glucose, glucose can't enter through this cell membrane structure here, but it can go through the protein carrier inside, from the outside to the inside of the cell. So what that phospholipid membrane structure allows us to do is allows us to be able to select only the things we want to come in for these protein carriers, and other things we don't want, we can just prevent from getting in. And it's very important to make sure we have a balance inside in terms of the substances we want to have inside this. If we don't have too many, we don't have too few, and the membrane allows us to do that. So another thing that's part of the membrane structure is your glycoproteins, and these glycoproteins here are outside. Usually they're not going through the whole of the actual membrane, or only part of the membrane. They have a carbohydrate attached. Glyco means carbohydrate, and this carbohydrate will be a receptor for the cell, so it allows it to be able to communicate with other cells, for example, and they're really important as well. These are common features of any fluid mosaic model of a cell membrane. That's basically how your zoomed in version of your cell membrane would look like. And down here is just another picture of how it would look like. So you've got your phospholipid bilayers. This is one phospholipid layer, and this is another phospholipid layer. That's why it's called a phospholipid bilayer, because it has two layers. It has these transport proteins, which would be these ones here. Right? So they allow things to go in and out that would otherwise be prevented from getting in. And it has these glycoproteins, which act as a receptor. These ones right here. So they allow communication with other cells. These are some features of the fluid mosaic model of a cell membrane. But what you should know is that they allow you to be able to pick just what you want. Right? They're selective with what they allow to come in, or semi-selective. They give you a nice boundary between the inside of the cell and the outside. It's like the cell membrane gives you structure. Make sure that things that are inside can't just escape outside unless they're not wanted. And that's where you find a lot of your lipids in your body as part of this phospholipid membrane structure. So far we've talked about things need to go into cells and also have to go out of cells as well. And now we're going to talk about how that actually happens in terms of some of the processes that are needed to make sure that things can move in and out of cells. First one you should know is osmosis. Osmosis is water moving from an area of low solute to an area of high solute, and it does so through a semi-permeable membrane. So for example, we said that your fluid mosaic model was a semi-permeable membrane, so that's just your cell membrane. Now if you were to, what you need to know here, a solute is just something that dissolves in water. So for example, this could be salt. Now if you have, so it says water, nothing else but water. Water travels from the area of low solutes. In this case, here we have low compared to here. This is low because that's only two of these salt particles to an area of high solutes. This here would be the area of high solute because we have more salt. So what we're going to find is water will travel from an area of low solute here through this membrane to an area of high solute. So water will travel this direction mainly. A bit might travel the opposite direction, but mostly in that direction. And then you would have done an experiment in class. For example, this here is your onion cell or onion strip. You put that in salt water. This here is salt water. So salt water means you're going to have high solute here. You have a lot of salt particles outside, only a few in the onion itself. So remember, water travels from an area of low solute to an area of high solute. So it's going to be low solute in the actual inside of the onion and high solute in the actual water around it. And that means water will leave the cell and go out into salt water around it. And what's going to happen to the cells of the actual onion is they're going to shrivel up. So that's an example of osmosis in action. You're going to have that shriveling up happen. So remember, with osmosis is water only moving from an area of low solute to an area of high solute. The other one you need to know about is diffusion. So this is right here is diffusion. Diffusion is a bit different. It just talks about a substance, not just water, it could be any substance, moving from an area of high concentration to an area of low concentration. So if you imagine this here to be a room, so these could be, for example, deodorant. This could be deodorant. You spray deodorant, which is just gas, and you sprayed it at one location, and let's say 2 minutes afterwards, those molecules of the deodorant will have moved from an area of high concentration where you started spraying, and it will fill out the rest of the room. It's going to be moving towards an area of low concentration, and what's going to end up happening at the end is it's going to be spread out. And you know that because the smell of that deodorant goes away after time, so it's going to be equalized eventually. So that's diffusion is a substance moving from an area of high concentration to an area of low concentration. So that's how osmosis and diffusion are different. Osmosis only deals with water moving, whereas diffusion only deals with any substance moving from an area of high concentration to an area of low concentration. And both of these, osmosis and diffusion, they occur in around cell membranes to allow things to come in and out of that cell. So one more thing you need to know about is when it comes to cell membranes is that we need to have a large surface area to volume ratio. And I'm going to show you why in a second. So the surface area is just whatever surrounds the cell. And remember, what surrounds the cell is going to be a cell membrane. So when we're talking about surface area, we're really talking about your cell membrane. Volume is basically the inside of your cell. So the volume refers to all of this space, which is your cytoplasm. It has all your organelles and all your chemical reactions. They all occur within the cytoplasm. So your volume is just your internal space, and your surface area is your actual cell membrane. Now it says we want to have a large surface area to volume ratio, and I'll talk about the reason why in a second, but I'll show you mathematically why as well. It's because if you have a more surface area, that means you have more cell membrane compared to the inside. What that means is, if for example all of these organelles here require glucose and different nutrients, if we have more cell membrane, it's easier for them to be able to get it. Everything's closer by, we can get things more quickly, and the cell needs to be working quite quickly. And any waste that gets produced can be moved out quite quickly as well, so we can remove waste quite quickly. And that means the cell is working at its optimum. It's removing waste, it's getting all the nutrients it needs, because it's all quite compactly, quite packed, well packed, right? And that's if you have a large surface area to volume ratio. Now if I've increased the volume a lot, I've also increased the surface area, but I've increased the volume more. What that means is we're going to have, we still need all these chemicals that come in, but because there's more volume, it's harder for all those things in the middle to be able to get it. And removing waste also isn't as easy as beforehand. So that means many of those organelles in the middle of that cell aren't going to be working as well as they were beforehand. So this is not ideal. Now, you might have done an experiment in class where you looked at the surface area to volume ratio, and you might have 3 cubes. One cube had 1 centimeter size, the other cube had 2 centimeter size, and the third cube might have a 3 centimeter size. Now, to get the surface area, all you have to do is that front face of the cube. In this case, for the first example, has 1 centimeter and 1 centimeter as the two sides. So we get the surface area of that cube. At that front face, we just do 1 times 1, which would be 1 centimeter squared. And then, because it has 6 sides or 6 faces, we times by 6. So that will end up with, we have 6 centimeters squared as your surface area. Volume is just 1 times 1 times 1, which would be 1 centimeter cubed. So that ratio is 6 to 1. We have 6, for every 6 centimeters of surface area, we have 1 centimeter of volume, 1 cubic centimeter of volume. Now here we have 2 centimeter sides, so surface area will be 2 times 2, which is 4, and then we have 6 faces, so 4 times 6 is 24. And volume will be 2 times 2 times 2, which is 8. So now we have 24 over 8, which is the same as 3 to 1. So now we have a smaller surface area to volume ratio. And last one, we had 3 centimeter sides. So we have surface area is 3 times 3, which is 9, times 6, which is 54. So 54 centimeters of surface area. And volume is 3 times 3 times 3, which is 27. And if we simplify that, that's a ratio of 2 to 1. So this has the smallest of all the ratios. The biggest is the smallest cube, has the biggest ratio of 6 to 1. And the smallest ratio is the biggest cube, which is 2 to 1. Now, if you did the experiment, if you put some iodine, put this in these cubes, could be gelatin cubes in iodine solution, what you'll find is the one that has the biggest surface area to volume ratio will be completely covered with iodine. The one that is in the middle will have quite a bit of the outsides covered with iodine, but nothing has reached the center, right? Because it has less surface area to volume ratio, which means it's harder to get things coming all the way to center. Whereas the biggest cube will have just maybe the outsides covered, but the internal sides hasn't been touched much. And that means that because it has the smallest surface area to volume ratio, it's hard for things to diffuse all the way to the middle. And if that were a cell, that means it wouldn't be getting all those nutrients it needs. So what I'm trying to say here is that cells are very small, because if you're very small, you have a very large surface area volume ratio, and that's where you need to be able to actually survive. So that's why we have cells that have such a small surface area to volume ratio.